For centuries, people imagined the citizens of ancient Rome as a single people with a single bloodline. They pictured families whose ancestors had lived beside the Tiber River for generations. They imagined Roman soldiers, senators, builders, merchants, and emperors all belonging to one ancient Italian population. But when scientists opened graves around Rome and recovered DNA from the people buried inside, that picture began to collapse. Some of the dead looked genetically similar to early Europeans. Others carried ancestry connected to the Eastern Mediterranean, the Near East, North Africa, and distant parts of the empire. Then researchers compared these Roman genomes with modern European populations. The result was not a simple match. The people who lived in Rome changed dramatically over time. Entire ancestry patterns appeared, became common, and then became less visible again. One of the greatest cities in history had not been built by one unbroken bloodline. It had been built by waves of people. And hidden among those waves was a genetic world that no longer exists in exactly the same form today. But to understand how that bloodline was lost, we must go back to a time before Rome had roads, armies, temples, or even a name. Thousands of years before the first emperor, the land around Rome looked very different. There were no marble buildings, no crowded markets, no giant arenas. Small groups of hunter-gatherers moved through forests and open grasslands. They hunted animals, collected plants, and survived in communities far smaller than the city that would later rise there. These early inhabitants carried ancestry, once found across large parts of Western Europe. Their lives left almost no written record, but their bones remembered. Inside the hardest parts of their skeletons, tiny pieces of DNA survived for thousands of years. That DNA gave scientists the first layer of Rome's human story. It showed that central Italy was never frozen in time. Even before Rome existed, people were already moving into the region, mixing with those who were there and changing the genetic landscape. The first great transformation came with farming. Several thousand years before the Roman Empire, farming communities expanded into Europe from Anatolia and what is now Turkey. They brought crops, they brought domesticated animals, they brought new tools and new ways of living, and they brought their ancestry. Across Europe, hunter-gatherer populations mixed with incoming farmers. Central Italy was part of this enormous change. The people living near the future site of Rome began to carry a combination of older hunter-gatherer ancestry and ancestry linked to early farming groups. But that was only the beginning. A second major change arrived before the Roman Age. During the Bronze Age, ancestry connected to pastoral groups from the Eurasian steppe spread across much of Europe. These were communities linked to wide grasslands north of the Black and Caspian Seas. Their movements reshaped the genetic history of Europe. Central Italy was affected too. By the time Rome began to rise during the first millennium before the Common Era, its population already carried the marks of several ancient migrations. The supposed original Roman bloodline was already a mixture, but the biggest surprise was still ahead. Scientists studying ancient Rome did not rely on legends or statues. They collected genetic material from human remains found at archaeological sites in and around the city. One major study examined 127 ancient genomes from 29 sites, covering approximately 12,000 years of history. The remains came from many different periods. Some belonged to prehistoric inhabitants. Others came from the age of the Roman Republic. Some lived during the height of the empire. Still others were buried after Roman power in the West had collapsed. Together, they formed something like a genetic time machine. Instead of examining Rome at one moment, scientists could watch its population change century by century. The process was difficult. Ancient DNA breaks down over time. Heat, water, bacteria, and chemical reactions destroy genetic material. Italy's climate is not ideal for preservation. Researchers therefore searched for DNA in dense bones and teeth, especially parts of the skeleton that can protect genetic material better than others. The samples also had to be checked for contamination. Modern human DNA can enter ancient remains during excavation, storage, or laboratory work. Scientists looked for damage patterns that are typical of genuinely ancient molecules. They compared hundreds of thousands of genetic markers. Then, they placed the ancient individuals beside reference populations from different regions. The results did not tell researchers the identity, language, or culture of a person. DNA cannot reveal whether someone called himself Roman, Greek, Syrian, Egyptian, or something else. But it can show broad patterns of biological ancestry. And those patterns revealed something remarkable. By the period when Rome was becoming a city, the people of central Italy already looked genetically similar to other Western Mediterranean populations. There was variation. But many individuals from the Iron Age and the early Roman Republic fell close to populations associated with central and western parts of the Mediterranean. They were not identical to modern Italians. No ancient group is exactly identical to a modern one. But the early Romans occupied a genetic position that would not have looked completely unfamiliar in later Europe. At first, this appeared to support the traditional image. Rome began as a regional Italian city, Many of its early inhabitants had deep connections to the populations of the Italian peninsula and nearby Mediterranean lands. Then Rome expanded, and everything changed. At first, Roman expansion was local. The city fought neighboring communities. It gained control of central Italy. Then it conquered rivals across the peninsula. After that came wars against Carthage. Rome seized islands. It entered Spain. It crossed into Greece. Its armies moved through North Africa, Gaul, the Balkans, Anatolia, Egypt, and the Near East. By the first centuries of the Common Era, Rome controlled a vast territory. Its roads connected distant provinces. Its ships crossed the Mediterranean. Its political decisions affected tens of millions of people, but Rome was not simply sending soldiers outward. The empire was also pulling people inward. Merchants arrived to sell grain, oil, wine, cloth, spices, glass, and metal. Officials came to work for the government. Teachers, doctors, sailors, servants, religious leaders, and laborers crossed the empire. Their children and grandchildren became part of Roman society. The city grew to a population that may have reached around 1 million at its peak, although exact estimates remain debated. Feeding that many people required a system stretching far beyond Italy. Luxury goods moved through ports linked to Asia and the Red Sea. Rome became less like an isolated Italian city and more like the center of a connected Mediterranean world. The ancestors of Europeans, Asians, Oceanians, and indigenous Americans all passed through that narrowing. Then each population experienced additional founder effects. The ancestors of indigenous Americans moved through several stages of separation and isolation. By the time people expanded into the Americas, they carried only part of the variation found in larger ancestral populations. But isolation also created new patterns. Genetic variants became common by chance. Others may have helped people survive cold climates, changing diets, high altitudes, or local diseases. This is why genetic diversity cannot be ranked as better or worse. Populations carry different histories. Africa contains the greatest overall human genetic diversity because modern humans have lived there the longest. Indigenous American populations preserve unique combinations formed through migration, Beringian isolation, rapid expansion, regional adaptation, and thousands of years of separate development. A major 2026 analysis emphasized both the complex history and the unique genetic diversity of indigenous American populations, while also showing how European colonization caused lasting losses of diversity. But this comparison leads to a darker part of the story. Some of the greatest changes in Native American DNA did not occur during the Ice Age. They happened only a few centuries ago. The genetic collapse after contact. Before European colonization, the Americas contained large cities, farming systems, trade routes, political alliances, and highly diverse societies. Then, ships arrived from across the Atlantic. They brought invasion, forced labor, warfare, displacement, and diseases to which many indigenous communities had limited previous exposure. Smallpox, measles, influenza, and other infections moved rapidly. The human cost was catastrophic. In many areas, populations fell sharply. These events changed the genetic record. European and African ancestry entered many communities through a mixture of consensual relationships, colonial settlement, enslavement, coercion, and sexual violence. The pattern differed across every region. Some indigenous communities retained very high levels of ancestry connected to pre-contact American populations. Others carry substantial European or African ancestry, while remaining culturally, politically, and historically indigenous. It cannot replace community relationships, law, kinship, language, and sovereignty. A DNA company may estimate that a person has ancestry from the Americas. That does not make the person a member of a native nation. Indigenous peoples are living political and cultural communities, not genetic categories from the distant past. And the effects of colonization created another scientific problem. Modern DNA may not perfectly represent the populations that lived in the same region 500 or 5,000 years ago. To reconstruct deeper history, researchers need ancient genomes. Who owns the past? Hay crossed a landscape that later disappeared. Their descendants moved from Arctic tundra to tropical rainforest. They reached mountains where the air was thin. They settled coastlines, deserts, plains, islands, and river valleys. They created societies so diverse that no single genome, language, culture, or origin story could ever represent them all. Modern science did not discover that indigenous Americans had a history. Indigenous peoples had preserved their own histories for generations. What genetics added was a different kind of map, a map of ancient separations, lost populations, unexpected reunions, and human connections stretching across the planet. The comparison with every continent did not reveal that Native Americans came from one surprising modern people. It revealed something far more powerful. Before the first ancestors entered the Americas, several ancient human worlds had already met inside them. Their DNA carried memories of countries that no longer exist, coastlines now beneath the ocean, and populations known only through fragments of bone. The map is clearer than it was, but it is still incomplete, and somewhere, Under frozen soil, inside a cave, or beneath the waters of the Bering Sea, another human genome may be waiting. When it is finally found, will it complete the story of the first Americans? Or reveal that the greatest surprise is still hidden? The country that disappeared. Today, Alaska and Russia are separated by ocean. During the last ice age, much of the world's water was locked inside enormous glaciers. Sea levels were lower. A wide region of land connected northeastern Asia with northwestern North America. This region is called Beringia. It was not simply a narrow bridge. At different times, it formed a vast territory of plains, rivers, dry grasslands, and cold steppe. Mammoths moved across it. Bison grazed there. Human communities may have lived there for thousands of years. A leading genetic explanation is known as the Beringian standstill model. According to this model, the ancestors of most indigenous Americans became isolated from other Asian populations during the Ice Age. Glaciers and difficult environments limited movement. The population remained separated long enough to develop its own distinct genetic profile. Then, as the climate changed, descendants of that population expanded into the Americas. Scientists have identified thousands of genetic variants that may have become common during this period of Beringian isolation. One genomic analysis reported more than 20,000 high-frequency variants consistent with genetic drift or selection during this long separation. Picture the scene. To the west stood the enormous spaces of Siberia. To the east, glaciers blocked much of the route into North America. To the south, coastlines looked completely different from those of today. A small human population survived between worlds. Generation after generation, they hunted, moved, raised children, and adapted to a harsh environment. They could not know that the land beneath them would one day vanish below the sea. But something even more unexpected had already happened before their isolation. Their ancestry was not entirely East Asian. Part of it came from another population. The proof was found beside Lake Baikal. The boy from Malta. In south-central Siberia, archaeologists discovered the remains of a young child. He had lived roughly 24,000 years ago near the village of Malta. The child is known to researchers as MA1. When scientists sequenced his genome, they expected another ancient Siberian profile. Instead, the results transformed the story of the first Americans. MA1 was not closely related to present-day East Asians. His genome belonged to an ancient northern Eurasian population with deeper connections toward western Eurasian hunter-gatherers. Yet he also shared a significant genetic relationship with indigenous Americans. The conclusion was extraordinary. The ancestral population of Native Americans had formed partly from an East Asian-related lineage and partly from a population related to ancient northern Eurasians, like the Malta child. A major study of the MA1 genome found evidence that ancestry related to his population contributed to the ancestors of Native Americans before their expansion into the Americas. This did not mean Native Americans were recently European. It did not support claims that Europeans founded the first American civilizations. MA1 lived thousands of years before the modern populations of Europe formed. His people belonged to an ancient branch that once spread across parts of northern Eurasia. Later migrations changed the genetic landscape of that region. The population itself largely disappeared, but its ancestry survived. Some of it moved west. Some of it entered populations in Siberia, and a major part traveled eastward with the ancestors of indigenous Americans. The first unexpected answer had appeared. When Native American DNA was compared globally, it did not connect to only one side of Asia. It carried the history of a meeting between Eastern and ancient Northern Eurasian peoples. That mixture happened before the settlement of the Americas. The ancestors of the first Americans were already the descendants of multiple journeys. But there was still a major problem. A connection found in modern DNA could have appeared much later. Scientists needed ancient American genomes. They needed to know whether this mixed ancestry was already present among the earliest known people south of the continental ice sheets. Then a child from Montana provided the answer. The child buried with red ochre. Around 12,600 years ago, a young boy was buried in what is now Montana. His body was covered with red ochre. More than 100 stone and bone objects were placed with him. Some were linked to the Clovis archaeological tradition, famous for its carefully shaped stone points. For decades, Clovis sites stood near the center of arguments about who first entered the Americas. Some researchers once believed the Clovis culture represented the earliest major population south of the ice sheets. Later discoveries showed that humans were present before Clovis, but the child's genome remained extremely important. Scientists called him Anzac 1. When his DNA was compared with populations around the world, the result was clear. He was more closely related to indigenous peoples of the Americas than to any population outside the Americas. He also carried the ancient northern Eurasian-related ancestry found through the Malta genome. That meant the mixture had already occurred before Anzac 1 lived. The Malta-related contribution was not caused by a recent historical arrival. It was part of the deep ancestral foundation of early indigenous Americans. But another detail changed the discussion. ANZIC 1 showed stronger genetic connections to many indigenous populations in Central and South America than to some northern North American groups. That suggested an early split. One branch became strongly connected to indigenous peoples farther south. Another contributed more to parts of northern North America. The settlement of the continents had not been a slow movement of one unchanged population. People were separating, expanding, and forming regional histories very early. Then researchers studied an even older genome from Alaska and discovered a branch that seemed to have vanished. The ancient Beringians. At the Upward Sun River site in Alaska, archaeologists found the remains of 2 infants. One of them lived around 11,500 years ago. Researchers recovered enough DNA to identify a previously unknown ancient population. They called this group the ancient Beringians. The name did not describe a modern tribe. It described a genetic branch revealed through ancient DNA. The ancient Beringians appeared to have separated from other ancestral Native American groups before the major division between northern and southern branches. Their genomes stood near the base of the indigenous American family tree.